Hello everyone. We're going to be working on page 10 and 11 of your note guide today, towards the bottom third of page 10 and on to page 11. So we're continuing to look at DNA being made into proteins. And this is a really big idea. Some people argue the biggest idea in all of biology when this was brought to light. We're going to talk particularly about transcription and translation today, so kind of breaking protein synthesis down into even smaller parts, if you will. And remember, the whole reason why we're looking at this is because this is how we are the way we are, and other organisms are the way they are, right? Our traits, in other words, are determined by these proteins. And so we're talking about proteins like insulin, right? And hemoglobin, melanin, things that we probably have heard of before. And these are the parts of our cells that make up, of course, us as humans or organisms. So, why do we need to decode DNA? DNA contains all the instructions that you need to sustain life. And it's kind of like a library, right? Certain things can't be taken out of the library, like the giant atlas or the huge dictionary, right? It's just there so this genetic material inside the nucleus is too large to be moved easily. So it stays there. So it needs to be helped along the way to be able to get the information from it to where it needs to go. And that's where the RNA, of course, comes in. So just a picture of it here in the upper left, you'd be looking at the nuclear membrane or nuclear envelope, and the DNA is inside there. And you can kind of look through the steps here, but essentially you're going from DNA on the far left, and it's kind of moving along this pathway that mRNA comes in and attaches to the ribosome, and this, of course, is what leads to these amino acids, or the monomers of proteins, being constructed into polypeptide chains, proteins. So how does this work? How do you make the mRNA? And think about transcribing, scribing, script, right? You're thinking, I hope, words. Trans means to go across, right? A transatlantic flight across the ocean, right? Across the Atlantic. And so somehow we're writing across from DNA to RNA. Very similar to DNA replication, except instead of getting a double-stranded DNA as our product, this time we're getting a single-stranded RNA, our mRNA. And it shouldn't surprise anybody that there are enzymes involved. Enzymes do a lot of the heavy lifting for cells, a lot of the work of cells. And so one here called RNA polymerase, poly we know means many, ase, of course, enzyme. RNA polymerase is an enzyme that puts together RNA, makes the mRNA for us. And it does that by attaching to an open section of DNA that's been cut open and copying a segment of that DNA. We normally think of that as a gene that it's copying. It gets a little more complex than that, as you can see here in the notes, but essentially that's what it is. We're writing from DNA into RNA. It's like looking at notes on a screen and writing them on a paper, right? You've transcribed them. So what are these proteins? Well, again, we're on the top of page 11 or so in your notes. These proteins are made up of amino acids. And I want you to think about this. There's 20 different amino acids. How many letters are there in the alphabet? Most of us probably know or could look it up easily. There are 26 letters in the alphabet. And if you start thinking about how many words can be made from that many letters in the alphabet, think about how many different proteins could be made from 20 different amino acids being put together in different formations. A lot of estimates suggest that nature hasn't even scratched the surface of the number of types of proteins that could be made and the vast number of things that these proteins could do. But nevertheless, they're made of amino acids, 20 of them, different ones, and they're strung together. And they start to take on different shapes because of the order of the amino acids. And another name for them, polypeptide. So here's an illustration of the process. Along the bottom there, kind of like a ticker tape going through, we have our mRNA. Remember, that came from the nucleus out into the cytoplasm, attached to the ribosome, which is another form of RNA, rRNA. And then these transfer RNAs, the tRNAs, are reading, right, as it goes here, and adding amino acids to a string, so to speak. Another illustration of it here at the top, you can see these amino acids with the 3-letter names there, right, abbreviations, strung together. Well, what will happen is some of them are polar, some of them are non-polar. They have charges in different spots, and that causes them to start folding on each other, on themselves. And when they combine with other ones, you get these big structures, these tertiary structures. Kind of think of like a sweater being put together, right? You've got the body of the sweater and the arms of the sweater, right? They're one big long thread of yarn, and then they're sewn together or put together in one final form. That's kind of what your protein is like. So how do we read this mRNA? Similar to how our words are made up of a set of letters, how to read DNA is the same, only simpler in that every word, if you will, in DNA is written in 3 letters called a codon. So every combination of 3 letters stands for something, right? It codes for a specific amino acid. So the codons there you can see are sections of 3. And there are ones like punctuation in our language that say start here, stop here, right? Think of like a period or a capital letter, that type of thing. To read codons, there are several different charts out there, and I apologize, I'm covering up the chart here a little bit on this, but there are charts on your in your book and in different places that are going to tell you the same information. But when you're looking at this chart, you're reading from the inside out. So as an example, you have CAC are the letters that we're reading in our mRNA. We go to the middle of our circle here and we find a C. Well, it looks like it's in the lower right-hand quadrant. And then we move out to the next rung, or the next concentric circle, which is an A, and then another C, and of course, that's the one I'm kind of covering up here, but maybe you can see it a little bit, called histidine. So that is the amino acid, this histidine, that is coded for, and that's what the transfer RNA, the tRNA, will go out and capture and bring back to sort of put this protein together. Okay, so another chart here, another way of reading it. It tells you here first letter, second letter, third letter, kind of starting from left and moving to the right, so maybe a little more intuitive. It's kind of like reading a sentence or reading a word. And if you look here, can leucine have more than one codon? Well, let's find it on our chart here. And yes, it can, right? If you look at leucine, it can have several different codes, right? It could be a C, CUU, it could be a CUC. And so, in other words, it would be kind of like a word having the same meaning, same idea here. But not all of them can be that way, right? Some words, so to speak, are unique and there are no other ways of saying it. And that's kind of how this tryptophan is. So in other words, codons can code for more than one amino acid, but in certain occasions they'll only code for one amino acid. All right, finally, translating. When you think about translating a language, it's the same idea, right? You're going from one meaning that you don't understand into a meaning that you do. And so that's what's happening here when the protein is being made. This is essentially the process that's taking place with the ribosome and the tRNA. The tRNA brings it in, so we're at the bottom of page 11 here. The tRNA goes out and finds these amino acids, and they actually have something on their other end called the anticodon. And if we know what the anti means, it means the opposite of. So if there's a CAC, right, they're going to have a GUG on their anticodon, and that tells them to go out and find whatever that codes for, right? If you remember, I think that was the histidine we talked about. It grabs that out in the cytoplasm and it brings it back to the ribosome and it attaches it to the polypeptide chain, and that growing chain eventually starts folding to become our protein. So we've translated from this mRNA that doesn't really mean much to the cell, right? Kind of a meaningless thing, although it's a blueprint, it's important, into something that's useful for the cell, the actual protein. Okay, so at the bottom of page 11 there, you'll do just a little practice. If you have, this is your DNA sequence, and it's already broken into threes for you, into the codon section, so to speak. First, you're going to transcribe it into mRNA. So you're writing across from the DNA into the mRNA sequence. That's your transcription process, writing across. Then you're going to figure out which amino acids are coded for. So you're going to translate it from its codon form into its amino acid and ultimately its protein form. So the two processes here kind of set side by side. Inside the nucleus, the transcription is taking place. A section of DNA is copied over in mRNA. That mRNA goes out in the cytoplasm, and this is where translation takes place. And translation is the process where the mRNA attaches to the rRNA, or the ribosome, and then the tRNA brings in the appropriate amino acids that the codons tell them to go and get, so to speak. And that's how you get translated ultimately into a protein, like hemoglobin, right? The one that carries your oxygen in your red blood cells. Incredibly important, right? We wouldn't be alive without it. Your body's cranking out hemoglobin all the time with this process. Another view of it there. And then there's your practice. If you think about what the mRNA sequence is going to be, again, transcribing it, you're copying it over. And then remember, we have to consider the anticodon. It's not as important, but we should be aware of it. And essentially, you're going to notice it looks very similar to the DNA, except where there's the T's. In DNA, we're going to have the uracil, or the U, in the tRNA sequence. But ultimately, when we go to translate, we're taking the mRNA sequence and we're going to our codon wheel or our codon chart, and we're translating from that to find our amino acids. So UUC would be phenylalanine, right? GAC is aspartic acid, etc. Okay. Take care, everyone, and we'll see you soon.